In this talk, we will learn about how to build phylogenetic trees. The discipline of systematics studies the diversity and evolutionary relationships, or phylogeny, among different organisms. These evolutionary relationships are pictorially depicted using phylogenetic trees. In case you were wondering the etymology of phylogeny, it arises from the word philo, which means tribe, and geny, which means generation. The phylogenetic trees can include not only organisms that are presently living or extant organisms, they can also include extinct organisms. Phylogenetic trees are made up of branches and nodes. The nodes denote ancestors. These ancestors could be direct ancestors, as shown for the chimps and humans, or they could be more distant evolutionarily. Thus, we can see recent as well as distant ancestors in the phylogenetic tree. The branches of a phylogenetic tree are represented by lines. Now, each branch is a hypothesis which predicts the evolutionary relationship among the different organisms in the phylogenetic tree. This is based on the information that is present with scientists. And as more information is available, if there is a need to move a branch to another location, it can easily be done in order to show the more accurate evolutionary relationships among the different organisms. Phylogenetic trees also show the presence of outgroups. Outgroups are taxa that are closely related to a group of interest, but they diverged earlier. So let us take the example of the sister taxa of chimps and humans. They arose from a common direct ancestor. Now, when we look at the lizard group, the lizards arose from an ancestor that is more distant than chimps and humans. And hence, in this case, the lizards are considered to be the outgroup. Similarly, if our group of interest is the group that includes the frogs, the lizards, the chimps, and the humans, then in this situation, the group of fish serves to be the outgroup because they did not arise from the same ancestor that gave rise to the frogs and the lizards and the chimps and the humans. The outgroup allows to root a tree. And in this instance, the root is going to be the part of the tree that shows the last common ancestor of all the taxa that we are interested in, which in this case were the frogs, the lizards, the chimps, and the humans. In the past, phylogenetic trees were constructed based on similarities among different organisms that were morphological, anatomical, or physiological in nature. However, we now know that the genomes of individuals retain evolutionary history because they are passed down from generation to generation. Thus, the descendants always tend to have some genetic information of their ancestors. Nowadays, DNA and protein sequencing tools are widely available, and hence they aid in creating more accurate evolutionary histories among different organisms. The goal now is to create groups that include an ancestor and all its descendants, and thus we are able to observe the evolutionary history and relationships of different living organisms. Groups that include the most recent ancestor and all its descendants are called as monophyletic groups. An example of such a group is the class of aves, which includes all birds. As seen in the phylogenetic tree, all birds arose from one ancestor. And when we have the phylogenetic tree that includes all the descendants, including that ancestor, it is a monophyletic group. Just to let you know that all the birds that are present today arose from an ancestor called as Neornithes. The goal today is to have taxonomic groups that should be monophyletic in nature. These monophyletic groups are now called as clades. A clade is a group of organisms that includes a common ancestor and all of its descendants, whether they are living or extinct. A clade may include only one species, as shown in the tree, or it could include a few of them. Clades can also be nested, and that means that we can have one clade that is part of another larger clade. We can also have situations where a clade can include thousands of species. The only unifying aspect of clades is that they all include the common ancestor and all of its descendants. Cladistics is a method where the primary criteria for classifying organisms is based on their common ancestry. Thus, it is a different way of classification. Cladistics based on identifying characters that are similar between different organisms. These characters can be the presence, absence, or loss of certain traits or features. For example, they can be organisms that have a body that shows bilateral symmetry, which means there is a line that can divide the body into a left and a right part that are mirror images of one another. Another trait could be the absence of hair. For example, fish don't have hair, they have scales. Similarly, we can have presence of hair as a trait. For example, mammals tend to have hair. The loss of a tail can also be a trait or a character, which in the case of humans, we know that our ancestor had tails, but we don't, and hence we lost the tail. Cladistics involves classifying organisms into groups called as clades based on the presence or absence of different characters. These characters can be derived or ancestral. Shared derived characters are those characters that are present in all members and evolved in the most recent ancestor. For example, when we look at mammals, like a leopard, all mammals have hair, and the character of possessing hair evolved in the ancestor that gave rise to these mammals, and hence the possession of hair is a shared derived character for the clade of Mammalia. On the other hand, if we look at the group that has reptiles as well as mammals in them, then the shared derived character is the presence of an amnion which involved in their most recent ancestor. If we go further along and look at a group that includes frogs, turtles, and leopards, or basically amphibians, reptiles, and mammals, then we can see that the shared derived character, which is the character that's present in all of them and evolved in the most recent ancestor, is the presence of 4 limbs, and hence they belong to the clade of Tetrapoda. These shared derived characters are also called as synapomorphies. Shared ancestral characters, which are also called as simpliciomorphies, are those characters that arose prior to the common ancestor of the group. So if we look at our phylogenetic tree and we focus on the group of mammals of which leopard is a representative, the shared derived character is the possession of hair which evolved in the direct ancestor of mammals. Now when we look at the presence of an amnion, which is usually present in eggs that are present in mammals, we're able to see that this character evolved in a more distant ancestor. And hence, this is a shared ancestral characters for the clade of Mammalia. Similarly, the presence of 4 limbs also evolved in a more distant ancestor, and hence the presence of four limbs is also a shared ancestral character for the clade of Mammalia. It should be noted that characters can be both ancestral and derived depending on the context. And so for this, let us take the example of these group of organisms where turtles and leopards are present. Now in this group of organisms, when we look at the common ancestor, it possessed or evolved the presence of an amnion in its eggs. Thus, for the group that includes turtle and leopards, the presence of an amnion is a derived character because it's present in the most common ancestor, which is the recent one. On the other hand, when we look at the presence of four limbs, then in this scenario, the presence of four limbs is present in a more distant common ancestor, and hence this character is a shared ancestral character for the group that includes turtle and leopards. Similarly, hinged jaws is a character present in a more distant ancestor and hence is also a shared ancestral character for the group that includes turtles and leopards. Let us now learn about the different steps involved in building or constructing a cladogram. A cladogram is basically a phylogenetic tree which shows the relationship between different clades. The first step is to identify the organisms of interest. In this example, we will look at the evolutionary relationships between a gorilla, a shark, a salamander, a tiger, a lizard, and a human. These organisms of interest will constitute our in-group. The second step is to identify an outgroup. These are species or group of species that are closely related to the in-group. In this example, the lamprey will be chosen as an outgroup. The lamprey is closely related to the in-group because it is a chordate. However, it is a jawless vertebrate, while all the other organisms are jawed vertebrates. The next step is to identify characters that will be used to build the cladogram. These characters should be homologous in nature and not analogous in nature. If analogous characters or features are considered, then we don't see descent from common ancestors. Let's take an example to understand this concept. Imagine that the character that's being considered is the ability to fly. Now, when we look at different vertebrates, which are organisms that have a vertebral column, and look at their ability to fly, we are looking at the animals that are birds, pterosaurs, and bats. Since we are looking at the ability to fly, it would imply that birds, pterosaurs, and bats arose from a common ancestor. However, when we look at the actual phylogenetic relationship among these flying vertebrates, it is very clear that the ancestor of the bat is different from the ancestor of the pterosaur, which is also different from the ancestor of the hawk. This way, we are able to see that these three organisms do not share a common recent ancestor. The flying character thus results in the formation of a group that is called as a polyphyletic group. Polyphyletic groups do not show descent from common ancestors. Thus, the ability to fly cannot be considered as a character while building a cladogram. In fact, any analogous feature or character should not be used while constructing a cladogram or a phylogenetic tree. The homologous characters that will be utilized can include gain or loss of features. For example, we can look at gain of jaws, hair, or loss of a tail, or gain of amniotic membranes or lungs, etc. Once we identify our characters, we now determine whether the characters chosen are present or absent in the different organisms in our groups. 0 indicates that the character is not present, while 1 indicates that the character is present. The next step is to determine if the characters are derived or ancestral. A character that is present in a group but absent in an outgroup is a derived character. For example, in the case of humans, being bipedal is a character seen only in humans and none of the other groups. Hence, this is a derived character for the group of humans. On the other hand, not having a tail is a character that is shared between humans and gorillas. Thus, this character is an ancestral character because it is a character that is present in the group of humans, but also in the outgroup, which in this case will be the gorilla because it does not show the character of being bipedal. When we look at the group that comprises of gorilla and humans, in this case, we are able to see that both of them share the character of not having a tail. Thus, the ancestor that gave rise to the gorilla and humans shows the derived character of having no tail. However, it shows the ancestral character of having hair because that is a character that is shared between tigers, gorillas, and humans. On the other hand, the lamprey is a group that has none of the characters that we are considering, and hence it is the true outgroup for all the groups that are under consideration. The final step is to draw the cladogram or the phylogenetic tree based on the different patterns that are shown for the characters that we have considered. And hence, it would look something like this, where we start off with a distant ancestor, and that ancestor gave rise to an organism that had jaws. Now, this jawed organism became the ancestor for another group of organisms that we are considering, which includes the shark, the salamander, the lizard, the tiger, the gorilla, and the human. That jawed ancestor gave rise to another ancestor that had lungs. This ancestor now was able to give rise to the salamanders and other organisms which had an ancestor that evolved an amniotic membrane. This ancestor gave rise to the group of organisms that include the lizard, but also an ancestor that evolved the presence of hair. This ancestor then gave rise to a tiger and other organisms related to it, but also an ancestor that lost a tail. This ancestor that lost a tail gave rise to the gorilla, but also another ancestor that became bipedal, which gave rise to humans. Once we have determined evolutionary relationships amongst different organisms, if we find a new organism, then to determine where its place is in the evolutionary tree, we use the principle of maximum parsimony. The principle of maximum parsimony states that the explanation that involves the fewest number of evolutionary changes will be favored. For example, if we want to figure where the frog lies in this evolutionary tree, we look at the different characters displayed by the frog. As we can see, the frog is similar to the salamander because it lacks an amniotic membrane, it has no hair, and it's not bipedal. On the other hand, we can also see that it's similar to the gorilla because it does not have a tail. So in this situation, where should we place the frog? Should we place it close to the salamander or close to the gorilla? Let us look at the scenario where the frog is closely related to the salamander. In order to understand this phylogenetic tree, let us look at the different evolutionary changes that happened. We started off with a distant ancestor, which gave rise to another ancestor. This ancestor gave rise to descendants that formed the salamander, and it gave rise to an ancestor that lost its tail, and then gave rise to the frog. So that is one evolutionary change. In another scenario, we're seeing that this ancestor gave rise to an ancestor that evolved an amniotic membrane, which is the second evolutionary change. This ancestor gave rise to descendants that gave rise to the lizard group, and also to another ancestor that now evolved hair. That is the third evolutionary change. After they evolved hair, the ancestor then gave rise to organisms like the tiger, but also another ancestor that lost its tail. That is the fourth evolutionary change. This ancestor then gave rise to the gorilla and the humans. Thus, there are 4 evolutionary changes that occurred in this phylogenetic tree. When we look at another phylogenetic tree where we keep the frog more evolutionary related to the gorilla, we are able to see that from that distant ancestor, we form an ancestor that evolved an amniotic membrane, which gave rise to another ancestor that evolved hair, which gave rise to another ancestor that evolved a tail. We have seen 3 evolutionary changes so far. This ancestor gave rise to another ancestor that underwent 2 more evolutionary changes, which is losing hair and losing its amniotic membrane. And thus we are looking at 5 evolutionary changes. The first scenario showed only 4 changes and hence shows the fewest number of evolutionary changes. And thus, from the principle of maximum parsimony, the phylogenetic tree that shows 4 evolutionary changes is the one that will be favored. Thus, the loss of tail in frogs and apes is purely coincidental and an example of homoplasy. Homoplasy is a phenomenon where similar features arise that are independent of descent and should not be used in order to find evolutionary relationships among different organisms. DNA sequences are now commonly used to construct phylogenetic trees. Let us now see how we can use DNA to construct a phylogenetic tree that shows the relationship between these 3 species of trees. In this example, we have the DNA sequences of the 3 species of trees, and we compare them to an ancestral sequence that functions as our outgroup. We are looking at 4 different positions in this DNA sequence. We can hypothesize that the phylogenetic tree can look something like this, or like this, or like this, or like this. And in each scenario, we are seeing that there are different evolutionary relationships among the different species. For the sake of simplicity, let us assume that the first situation does not exist, and hence we have to choose whether tree 1, 2, or 3 shows the correct evolutionary relationship. Now let us consider the first position of the DNA sequence and look at what all changes would have happened related to the ancestral sequence. So for tree 1, we are able to see that there was 1 change from an adenine to a cytosine between species 1 and 2. Similarly, we can show these changes in tree 2 and tree 3. And we can very clearly see that in tree 2 and tree 3, 2 evolutionary changes occurred. Let's do the same thing for the second site. And in this scenario, when we look at all the different trees, we can see that there is 1 evolutionary change for site 2 in tree 1. There are 2 evolutionary changes for that same site if we consider tree 2. And similarly, there are 2 evolutionary changes at that site for tree 3. When a similar analysis is done at position 3, we are able to see 2 evolutionary changes in tree 1, 1 evolutionary change in tree 2, and 2 evolutionary changes in tree 3. For position 4, we are able to see 2 evolutionary changes in tree 1, 2 evolutionary changes in tree 2, and only one evolutionary change in tree 3 for that fourth position. When we compared the different evolutionary changes that happen in each tree at those 4 sites of DNA, we were able to see that tree 1 shows 6 evolutionary changes in total, while tree 2 shows us 7 evolutionary changes, and the same 7 evolutionary changes is shown for tree 3. Using the principle of maximum parsimony, we can now agree that tree 1 is going to be the most favorable tree because it shows the least number of evolutionary changes. Thus, in this talk, we learned about phylogenetic trees and how to read them. We also learned about clades and how they are monophyletic groups. We then learned about the steps involved in building cladograms, whether they take into consideration different morphological, anatomical, or physiological characters, or they look at molecular data like DNA. We also learned about the principle of maximum parsimony and how it can be applied to build phylogenetic trees.